So I give you some examples on haplosufficiency and haploinsufficiency. So you will see that when we think haplosufficiency, we are thinking complete dominance. When we think of haploinsufficiency, then that would mean that not having this second allele doesn't, it impacts the phenotype in a way that you are not going to see the phenotype of the dominant allele. So it will be a mixture of them. And then I was giving you the example of an enzyme. That's probably where we're going to start. And then I will go into gene expression. So that's the part that I'm going to go fast. This one is the dominant one. If you remember that whenever there is the heterozygote, this guy is yellow because, so being yellow means that there is this degrading chlorophyll happens. So that allele that we're talking about is really the allele that encodes this enzyme here. And having at least one allele that encodes the functional enzyme, it makes enough enzyme to degrade all the chlorophyll. If this was incomplete dominance, then you would see some P that would be not really yellow, not really green, which would indicate that you would need more units of this enzyme to completely degrade chlorophyll. So that would be the case of incomplete dominance. In complete dominance, there is haplosufficiency, which is if you have at least one allele of the functional enzyme, then you will see the phenotype of that dominant allele, which in many cases is the functional enzyme. Okay, so in incomplete dominance, I stopped, I think, at the part where I'm telling you about haploinsufficiency, and I was explaining why you having a second allele would interfere with could either interfere with the action of the functional allele, or you don't have enough of that enzyme to do the job, to get to, so for example, the flower is red because you have an enzyme, that's the chalcone synthase, that makes the red pigment. This flower is white because this enzyme is broken. It doesn't make the pigment. And then in here, you have something pink because you have one that has the functional chalcone synthase, and the other one that has the non-functional chalcone synthase. So you don't have that many workers making the pigment, and that's how you end up with somewhere in between. And then you will see in homework 2, I will explain to you even in more detail on gene expression in here. So it will encompass the gene expression and something similar to this. So the chalcone synthase is a dimer, so it has 2 units. So this part is encoded by one allele. This part is encoded by the other allele, or the same allele that got transcribed or translated twice. And they come together, and this is your functional enzyme. And this one would be our non-functional enzyme, which I changed the color just to make it different so that you know, like, okay, this one has some mutation, maybe it's in the catalytic domain, maybe it's in the dimerizing, maybe they don't come together, and that's how you don't get this very functional enzyme. For whatever reason, this allele makes an enzyme that is not functional. And then when you have an enzyme that is partly functional, then you get that, or if it was a monomer, maybe you just don't make enough of the monomer to make enough pigment. So in this case, this enzyme is partly functional in the case of, specifically for snapdragons. That's what that flower is. Okay, so here's where we stopped on Wednesday. In the case of haploinsufficiency, so I'm representing as a box the DNA sequence, and I'm representing it as the orange one, the allele that encodes a functional subunit. So you could have the one strand of DNA, and you could make 2 mRNAs out of it, and each mRNA be translated once. That's not necessarily true. You could have one mRNA that is very stable, and it gets translated multiple times. So it's in the cell is not really a 1-to-1-to-1. It depends on the stability of the mRNA, depends on RNAi, so there's more to the story than what I'm telling you. But in this case, this one copy of that gene gets transcribed into this mRNA, and then each of the mRNAs is translated, and you make the 2 subunits, which make an active enzyme. And then in the heterozygote, you have the one functional copy and the other one that is not functional. I put this red bar here to signify there's an early stop codon, so there's some mutation that makes this protein not to be the entire subunit as it would be here. So the mRNA is still the same length, but when this gets translated, the protein is only this long, so it's missing this chunk. So it makes some subunit that is different from that. Okay, so if the alleles come together, they make the functional ones make a functional protein. If those ones don't come together, or they come together, they don't make a functional protein. Maybe it doesn't dimerize. Maybe they don't actually bind to each other. And here, maybe these 2 parts bind to each other, but not very efficiently. So this enzyme doesn't work that well. Okay, so this would be the case of haploinsufficiency. And then you could think, okay, but how about if it was a case of haplosufficiency? How does the non-functional allele not impact the activity of your enzyme? So here's an example. Here's your functional one. This one is your non-functional one. And in this case, this guy has an early stop codon, but this mRNA gets degraded in the cell. So if this RNA gets degraded, then the subunit, the protein, doesn't get made. So this is one example of haplosufficiency where the functional copy gets made, and then you would have enough of these proteins to confer the phenotype of the dominant allele. And that this one doesn't get made, so it doesn't interfere with the action of this guy, as it does here. Okay, so up to now, I think it's kind of review from Wednesday. Do you guys have questions?